Erwin starts with his theoretical hands-on, a reminder that tomorrow and the day after, you need to take your laptops because there's going to be simulations, numerical experiments during the hands-on sessions. Thanks.
Thank you. So the actual hands-on will be tomorrow. So today is more of a lecture with a little part of hands-on. The reason is we forgot to communicate that you need your laptops for that. And the hands-on will be also done by my students here in the front row. I hope you still have some energy left after such a long day of all exciting lectures and information. And I kind of want to recapitulate a theme that we heard today in several forms, and which is a quite important theme in biology, namely how molecular is molecular. And that's not such a trivial question for biological systems, because after all, you have genes, you have proteins, there are specific genes, there are specific proteins, there are specific protein reaction networks, and it's a priori not clear how much of that you can coarse-grain and how much of that you cannot coarse-grain. So again, question is, how molecular is molecular? How detailed do we need a model to be in order to actually describe the biological system that we are interested in specifically, okay? You heard me talk about a very general type of model in my second lecture, which is this two-component system. And this two-component system is really of a lot of value because it teaches you the basic principles, and maybe I just recap what the basic principles are. One is really that you have a mass redistribution instability, and the flow and redistribution of protein mass is the driving force behind the pattern. Okay, that's the main point. Now today, I would like to do with you a small hands-on, which is setting up a molecular model. So really going from a reaction scheme to an actual diffusion reaction model. Second theme will be actually, after you've set it up, trying to understand the detailed molecular model. And of course, when I say understand, I mean understand in my sense, in the sense of a theoretical physicist that would like to see the principles that are underneath. The third point will be, as a good theoretician, you have to test your model. You have to test whether it's actually true and can explain concretely, specifically experimental data. Not just from a phenomenological point of view, but really as a function of parameters, as a function of different environmental conditions, etc. Now in biology or biological physics, there's various ways of testing theories. One is to test a theory in a reconstituted system, and with that we mean a system which is taken out of the actual biological system and reconstituted outside of the organism. That's called a in vitro system. And I'll do that. And then also show you how you can test this detailed molecular model in the actual living system, so in vivo. And for both, for the last two parts, I had over the years many people I worked with. So for the in vitro system, we worked a lot with K.S. Decker from the TU Delft and Petra Schwille from Munich. And for the in vivo testing, I recently worked with Sukun Yun from UCSD. So here we go. This is what I wanted to show you, that in addition to having the biological system that you want to study, so this one here, where you have the min oscillation, I don't know whether the movies run or not, you can actually take the two proteins, min D and min E, out of the E. coli cell, put it in a container, in a Petri dish like so, where the membrane is on the bottom, and you have some fluid on top, and the fluid mimics the composition of the cytosol, not in full detail, but to some degree. And then you have the height of this volume. We put just the two proteins in. And what you get, and this is really a beautiful paper from 2008 from the Schwille group by Martin Lohse at that time, what they found is that these proteins that show oscillations in the living system, they show these kind of spiral waves in the reconstituted in vitro system. And they can label the different proteins with fluorescent dyes. So mean E is labeled with a red fluorescent dye, mean D with a green fluorescent protein. And here you can see how the different patterns look like. They look very similar, but in the end, they're kind of overlaid and similar as in the actual system, the min E is kind of chasing the min D, right? What you should notice also, and this is not a minor issue, this is about a micron in length. That's a size of a living cell, and I'm not sure you can read it. There's about 100 micron or 50 micron here on this picture. So it's very, very different length scales that you encounter here. And it's not clear, actually, whether the phenomenon that you see here is the same phenomenon that's driving this. It's the same underlying biomolecular network, but as we know, when you change parameters, when you change environmental conditions, there's bifurcations, there's phase transitions, etc. So whether this is in the same part of the phase diagram as that one is not clear, right? But what's clear is that we do have quite a range of experimental data available to check and test the theoretical ideas. All right. Now, this is the in vivo system put on a computer, and we're going to come to the explicit modeling for now. What I want to tell you here is the story behind that. So a story in words, why there are those oscillations. I think I explained that to you. This is the cyclic flux of proteins between the cytosol and the membrane. And it's always such that the min D have a positive feedback to accumulate on the membrane. The min E is coming towards the min D, and then it's hydrolyzing the min D and kicking it off the membrane. So what you see here, and I guess I rerun the simulation, is that you have these pole-to-pole oscillations. They're more like shifting a pattern to the right and then shifting the pattern to the left. So it's more like swimming, pushing things to the right and left. And what happens there is at a pole, you have the min D accumulating first. That's kind of the first thing that happens. And then the min E comes after and kind of puts a ring at the back of this pole of min D proteins, and it's chasing it and pushing it off the membrane. And that's what you see. You form a polar cap, the min E is chasing it off, etc., back and forth. Now, when you think about this, there is one element here in this min E, min D dominance that's alternating, which has to be the case in order for that thing to work in the first place. Okay, and this is now heuristic arguing, but those things are important. Now, imagine you have much, much more min D proteins than min E proteins. Okay? Then, do I have it here? Then you can form these patterns because you can have this dominance of the min D proteins, and after that, the min E can kick it off. If the inequality sign that I show here is the other way around, then you have lots of min E, meaning you have a huge amount of the antagonist that's kicking off the min D from the membrane. And that means, in this type of model, you would not be able to get a pattern because you would not be able to actually form a pattern in the first place. There's too much of the antagonist available and kicks you off the membrane. Okay, so keep this in mind. This is a very strong prediction of the model that this inequality has to be satisfied in order for the pattern to actually form. This has to be tested experimentally. Now let us go to construct the model, and I'll guide you through. So, this is the reaction network that we have. And what we want to construct is this. So we want to construct the diffusion reaction equations on the membrane, in the cytosol, and the boundary condition that's connecting the two diffusion reaction equations between the two compartments. And I would like to do this in a small hands-on together with you. So and I hope you're still able to contribute a bit to that. So let's look at the model. This is the reaction scheme. The line is the membrane, and the rest is the cytosol. So let's write down the dynamics in the cytosol. Right, so how would we do this? First, we should specify the kind of components that we have in the cytosol. We do have a min-D protein, and let's call this here a CD, right? The D, the capital D, indicates that we have this min-D protein. And now I'm adding another D, saying that this min-D protein is in the ADP state, so in the passive state, right? That's one player. The next player that we have is also a min-D protein, but it is now in the active state with the ATP boundary. So that step from the right, the blue dot, to the green dot that we have. Okay? And you also have the min-E protein that you can study here. Sorry. Now, what we want to know is what is the time evolution of these three concentrations. That's the only players I have. Well, what's kind of easy, right, is to say there's a diffusion coefficient in the cytosol, and there is a Laplace operator acting on this. And of course, what I'm omitting here is the arguments of space and time. Space is here the 3D space of the cytosol, and time is just time. I have a choice here, right? I have all these diffusion coefficients, and I'm writing them here now in the form that I use DC for all three of them. That's of course not correct, strictly speaking. These proteins have different size, and because they have different size, they have a different friction coefficient, and because they have a different friction coefficient, they have a different diffusion constant. As we heard, there's a relation, the Einstein relation, relating friction coefficients with diffusion coefficients. But let's simplify for now and take all the diffusion constants to be equal. And then you do have here just the Laplace operator for my three components in the cytosol. Now, so what are the reactions that we have? Look at the min-E. Is there a reaction in the cytosol? There's none. It's just there, it's just diffusing, so that there's no reaction here. And let me write this as a 0. So there's nothing that I can add here to the equation. For the min-D, I have a reaction, and this reaction is called nucleotide exchange. I exchange the diphosphate to a triphosphate. Okay, so here we have nucleotide exchange, and that happens with a rate lambda. That's the name of the reaction constant. So what should I put here on the right-hand side? Any suggestion? Come on, it's not hard. Yeah?
And the second equation, d to t,
Yeah?
you multiply it.
So here I have terms, right? Some reaction terms on the right-hand side.
Yes. So
It's like in the master equation that we heard today. There's gain, there's loss, right? And the nucleotide exchange, if you do have a inactive protein and it's activated, it's lost in the first equation. So you have minus lambda CDD. Okay, and one's loss is the other's gain. This gives rise to a term which is plus lambda CDD. Okay? That's all that happens. And that's beautiful, right? If you look at an equation like so, then you immediately realize this is linear. And if it's linear, and that's the beauty of the cytosolic dynamics, you can actually solve it. And you can solve it in different spatial, even analytically solve it in different geometries, because that's just a linear problem, an eigenvalue problem. And it depends only on the geometry of the system, what kind of eigenfunctions you have. Okay? All right, so that's for the cytosol. What about the dynamics on the membrane? How would we write down that? And for this, I'll help you. We do have here a range of different processes. One is attachment and recruitment of the min-D. So it is, when you look here on this reaction scheme, it's the process where you have the min-D ATP attaching, that's the green arrow, and there's positive feedback. This is this looped arrow that you find here. Okay? And let's call this a reaction plus, the plus, it's just naming here. I put here to indicate it's going to the membrane. There's something added to the membrane. And the min on the membrane, just the convention that I use, because you have to use some terminology here, is a lowercase d, and the uppercase d is reserved for proteins that are residing in the cytosol. Okay, so if you do have that, then what should you write down when the basal attachment rate, so Kd is the attachment rate of the cytosolic min-D to the membrane, and the recruitment rate is Kdd because it connects the two proteins, the one on the membrane and then the one in the cytosol. So what should I write now according to mass action law? We had this. What is the corresponding rate? What's the rate for the spontaneous attachment of the min-D proteins? To what is it proportional?
To the concentration of
course, to the concentration of the proteins, so CDT, but where? at the membrane, right? Right at the membrane. It's not the concentration somewhere in the cytosol. There's a spatially extended cytosol, and all that matters for the attachment rate is how many do I have right underneath the membrane. So it's at S, and this is just indicating the surface that we have, the membrane surface. But there's nothing else. This is what you have for the spontaneous attachment. What about the recruitment term? Recruitment means, right, this is all language here, and you have to learn how to translate those kind of language into equations, that you have feedback, already present protein on the membrane facilitates the attachment of a protein that is in the cytosol. So what should it be proportional to? Same question. The concentration on the membrane, right. So this is it. Right? So you see, that's not hard. It's really just mass action law that we're using, and we have to write down the corresponding rates coming from that. Now, what about the recruitment of the min E by the min D ATP that's residing on the membrane? And I call this RDE plus to indicate that this is recruiting of min E through min D. What should I write? I'll give you the rate, because that's the per capita rate, KDE. What should it be proportional to? Don't be shy, it's very simple.
Is it CE times MD?
It's MD, yeah, because those are the guys that do the recruitment, right? You need proteins on the membrane to recruit it. And what are you recruiting? You are recruiting the min E proteins that are right at the surface. Okay, so the last one is hydrolysis, and that I call RDE. And I put a minus here because hydrolysis is the event where you have the dimer, this one here, the min DE dimer, and within the dimer there's hydrolysis happening such that you have switching off of activity and going back to the cytosol. Well, this is then another rate that I call KDE, and I need another concentration because this is now proportional to the concentration of the heterodimer made out of min D and min E. And that's it. So there are three processes happening, and these three processes, according to mass action law, are written down by these equations. It's quite key here. The models that we used in the two-component model, they were cooked up. They had some kind of nonlinearity, and I didn't really explain to you where the nonlinearities are coming from in detail. I just put them there to have a model that kind of works. Here, this is a molecular model, and the parameters that you have here, you can measure. At least you could measure if you could convince your experimental colleagues to actually make the effort in measuring those rates. They usually don't. That's the pity. Okay. Question? Yeah? It does, but that will show up then in the equation. These are the basic processes, and those basic processes then I have to write as equations, and there this will appear. I will write them down for the, that's still missing. These are just the elementary processes, and I didn't write down the equations yet. Yeah, thanks, Veronica. Yeah.
if instead using the concentration of the proteins close to the surface of the membrane,
Yeah.
if you use the concentration of in the whole cytosol,
Yeah.
would that be wrong?
Yes. That would in general be wrong because, as you can see from these processes, they are sources to concentrations in the cytosol. So you have a source degradation process going on from the membrane to the cytosol that defines a length scale. So you will have a gradient of the concentration of the MIND and MIN-E proteins that goes from the membrane to the cytosol. So you have spatial structure. And that's quite important. The cytosol is not just a reservoir. It has a structure on its own, and that structure on its own is relevant. Not always, but in general. It would be not relevant if the processes are such that diffusion is so fast and the reaction rate is correspondingly such that the length scale is basically huge, right? Then you would have a homogeneous reservoir. Yeah? Okay, now to setting up the equations. So let's write down the dynamic equations. So what do we have? We have on the membrane, we have just two things. We have the MIN-D proteins and we have the MIN-DE dimer. Okay, they will obey some diffusion equation. Oh, sorry, this is nonsense. dm d squared sigma md dm d squared sigma mde. So I'm making the same assumption as before. I say the diffusion coefficients on the membrane, they are the same for the two species. That's an approximation, but that's something we live with. Then I write here nabla squared sigma and not nabla squared. Why? Well, you have diffusion on a membrane, right? This is a two-dimensional surface, and sigma is indicating the coordinates in that surface. And if this surface is curved, or even more interesting, if this surface is curved and fluctuating, then you have here, of course, the Laplace-Beltrami operator and not just the Laplace operator. Okay? That's something I wanted to note. We don't really dive into this more than that. Now, what of those reactions that we have here are contributing on the right-hand side for the MIN-D protein? What do we have there? So make a pick. Is this important? Right, this is the recruitment term, and I have to add it with a plus. So plus RD plus. And this depends on all the concentrations in the membrane and all the concentrations in the cytosol at the surface. The expression I wrote on the left-hand side. What else appears here? This is a gain process. You gain by recruitment and attachment. What else do we have that contributes here on the right-hand side? Still awake? This one? Here, the MIN-D is recruiting the MIN-E. That's not contributing to the MIN-D itself. But what about this one? This describes hydrolysis, so it's a loss of the MIN-D proteins that you have. Okay? So you have minus and also the same kind of arguments. So what do we have here for the MDE? to make your pick. So the recruitment of the min D makes a dimer. So it's this term here. Oh, sorry, I said something wrong here. I was fooling you. So I was fooling you. Well, here in the second equation is plus. In the first one, it should actually, I said something stupid and wrong. This is RDE plus. It's not the hydrolysis. I said something stupid. I was wrong. So you were right. I said something stupid. It's the hydrolysis event. That's here a loss event because it turns the min D into a min. It's the recruitment of the min E that makes the min D in a min DE dimer, and that's a loss for the min D monomer on the membrane. That's the right way of saying it. Okay, so this is what you have, and now here, it's what I tried to say earlier. This is the hydrolysis, because now the hydrolysis event destroys the min E DE dimer that you have on the surface and makes it go into the cytosol. Okay, so those are the equations that you have on the membrane. Now, Veronica is not quite happy yet because the processes she was talking about are not there yet. Because this is just what's going on the membrane and what's going on in the cytosol. I still have to have the boundary condition. So, okay, so I'll help you. So here I'm counting on the right-hand side, how do you gain and how do you lose in the reactions? How do you gain a min D protein on the membrane? You gain by recruitment and attachment. That's this term. That's this one. Okay? How do you gain? Well, that's it. How do you lose? If you have a min D on the membrane and it's recruiting a min E, it's transformed into a min DE dimer. So that's a loss term for the min D concentration. It's a gain term for the min DE concentration. That's why you have this here. And finally here, when you have hydrolysis, the min DE dimer on the membrane goes away because it goes into the cytosol. Now, the last thing we want are the boundary conditions. And the boundary conditions you should visualize like so. You have a membrane, you have the cytosol, and there's currents. There's a current coming from the reactions. For example, the current coming from the hydrolysis reaction that gives rise to the flow of min DE dimers into min E and min D. And that flow has to equal the diffusive flux. That's a continuity equation that you have to have in the system. Otherwise, we'll gain or lose particles. So the way you write that is that the diffusive flux, which is easy to formulate, this is minus dc, the gradient perpendicular to the membrane of all the different cytosolic contributions, so this is a vector, at the surface, that's the diffusive gradient, that's given by the reactive currents that I write as F of M and C at the surface. So let's look at the different terms. There is a diffusive flux for the min D in its inactive state. Okay. What's the reaction that gives rise to a diffusive flux of an inactive state? Look at the model. The hydrolysis produces what? Min E and inactive min D, right? So the term I should write on the right-hand side is the hydrolysis process, right? So this is what you have here. This is RDE minus. And that's one of your terms, Veronica. It's the hydrolysis that gives rise to that flux. So hydrolysis produces inactive min D. Now you have a diffusive flux for the other player that we have in the cytosol, and that is the active min D. Okay? So how is the active min D flux? Where's this coming from? Is it coming from something that you get from the membrane or something that you lose to the membrane? What's lost to the membrane? The recruitment, right? The attachment, okay? So this is minus, because it's lost, minus RD plus. This is the attachment of active min D. And finally, we do have the diffusive flux of the min E proteins right at the surface. Okay. Well, there's now a combination of two things. You gain, again, by hydrolysis, because that brings particles into the cytosol, but you lose by recruitment of the min E from the cytosol to the membrane. Okay, so what you have is you lose by the recruitment of the min E, and you gain by the hydrolysis of the min E dimer. So this is hydrolysis and recruitment. Of min E or by min E. So we're done. You're through. Don't have to suffer anymore. This is how you get those equations. And it's all just biomolecular reactions and bookkeeping and gain and loss processes. But now we have the challenge, right? There's the lambda. There is this rate, and that rate, and this rate, and that rate. This is 1, 2, 3, 4, 5. 5 rates, 5 parameters. This one is measured. This is 5 hertz. This parameter lambda, the nucleotide exchange rate, has a bound. People know it's about 5 hertz. For the other parameters, super hard to actually measure them, because in experiments you would need to really control the actual concentration of the proteins and then make local measurements of how these biochemical reactions take place. Good, so you don't have that, but you still need to make simulations. You still need to make something out of this. So you have to come up. Pardon? Kind of. If you look into various different publications, these numbers are actually all over the place. And there is, of course, now difficulties in, we don't know anything about those parameters from measurements. You can just, argue heuristically whether the numbers you put down somehow make sense in a biochemical context. Yeah. And then in addition, if you now fit the parameters to your data, if the model is wrong, the fit is wrong, right? Because you try to adapt the data to the particular measurements that you make. All right, so here, this is kind of a set of numbers. Oh, I forgot to say that there's a diffusion constant here, and they are measured, right? The diffusion constant is something that you can really measure by single particle tracing. So you look at single particles, like the muscles that we saw, and you track them, and then you infer the diffusion coefficients, or you use something like FCS, fluorescence correlation spectroscopy. This is another method you can determine those parameters from, and then you know the diffusion coefficient in the cytosol is something like 60 micrometers squared per second, and you can do the math for your own. You know, how fast does the cytosolic volume mix? Ask yourself, right? One micrometer in diameter, diffusion coefficient is 60 micrometers squared per second. That's fast, right? This is fast on the second time scale. It's a very fast mixing process. On the membrane, this is basically very, very slow. And diffusion on the membrane is not such a big effect. Most of the mixing happens in the cytosol.
Can I ask a question here? So probably the students also, like pedagogically, so obviously the concentration on the membrane is per micron, the density per micron squared.
Yeah.
And then in the volume, in the cytosol,
Exactly.
you use the cubed. So, but
That's in there.
Yeah, and I can understand that you can talk about a total one when you think about a cross-section like through the bulk. But when you do that conversion, do you go from a...
Yeah, you do.
Yeah, okay.
You do. So you really, I mean, there's a concentration, a volume concentration, there's a surface concentration. One is per micron cubed, one is per micron squared, and you have to be serious about this. And we are serious about this.
And that also means that your constants will have to be correctly dimensionalized. For
exactly.
that.
This is...
And many people do this wrong.
Yeah, I know. This is a part where you can make a lot of errors. Yeah.
Quick follow-up question then.
Yeah.
What's the fraction of the mean that's on the membrane at any given time?
It's a low number.
It's a low number.
It's a low number, so you should think of these things as a gas. It's a diffusion reaction system, so most of what determines the dynamics is either diffusion or biochemical reactions. We heard about phase separation. That's not such an important term in these systems, right? There's no attraction or repulsion. There's some indication for the MIN system to be clear that they have seen some little clusters of MIN-D proteins, and it's not clear at the moment where they come from.
No, I meant the partition between cytosol and membrane.
What do you mean?
What's the fraction that is in the cytosol versus the fraction in the membrane?
Depends, right? So it's fluctuating back and forth. And when I show you movies, you will see that there's things attaching and detaching and attaching and detaching. And you can tune how much you have on the membrane or off the membrane by the cytosolic volume that you have. And you can work out the numbers. I don't know them by heart. And that's different for the in vivo system and the in vitro system, and it depends on how much you have in the cytosol. Yeah. Yeah.
Just to comment on this, like in general, you can, if you're looking at a different system, which is in the MIN, you can look at what's on the membrane because they GFP it, so you can measure that. And sometimes they take the whole cell and grind it, and they'll tell you so much is of this protein. So you can calculate out the fraction indirectly. So this is, every case will be different, but the fraction is. But it's important to just look through the literature to try to find those numbers, because then you might not know the number, but you could get the parameter, so you get the fraction sort of correct.
Yeah, so on that, if you do experiments, then you have to measure the number, and a typical tool is to label them fluorescently. And if you label them fluorescently, you can possibly count. Count in the sense that you measure concentrations or fluorescence intensity, and you have to transform the fluorescent intensity into numbers. That's a non-trivial exercise because this is a proportionality factor that's not so easy to get. If you're really serious about that stuff, you could do mass spec and whatnot to measure this in glory detail. Yeah, but thanks for mentioning all this. These are critical experimental questions if you want to be quantitative, and they play a role when I come to the in vivo system later on. There people went at lengths to really measure what the number of min-D proteins and min-E proteins is on the membrane. All right. When did we start? At about 5, right? Yeah. Other questions? So I'll skip the understanding of the model. Let me go right into, or do we do have time? We have 2 hours, right? I have to, up to 2 hours. Okay, then I do the understanding because that's dear to my heart. So, it's also, of course, always a question, what do you mean by understanding? This is my version of understanding. And that's in terms of this concept that we learned yesterday about redistribution of protein mass. There we had a single protein, and we had one axis. Now we have 2 proteins, so we have 2 axes. One of those axes, N1 and N2, they correspond to the total concentration of the min-E and the min-D proteins. Now, once you have here this phase space of min-D and min-E, and you assume it's well mixed, so as we did for the 2-component system, what can you do? You can do a bifurcation analysis. You can study what kind of fixed point structure do you have as a function of these 2 control parameters. So let's do that. Let's try, and that I call control space, just to be clear in names. You pick a certain concentration. The yellow dot is one particular realization of your system. And now you do a bifurcation analysis in that control space of the concentrations. And what you will get is something. And what you get for the min system is basically this. You get part of this control space being stable, so small small fluctuations of whatever they are, they will not change the stability of the system. And there will be a range of parameters, meaning the concentration of these total min-E and min-D concentrations, where the system is oscillatory unstable. So it has a limit cycle oscillation. Okay? Now you can move things back and forth. So you can move, this is not animating the right way. You can move this total concentration into the different parts of your phase space. But now if you leave it here, what was one of the messages that I tried to convey when systems become unstable to small perturbations? Mass redistribution, okay? That's key. So this means this little spot here, which is the average concentration, once the system is unstable, it will spread out, like explode. So it's gonna spread out through a lateral instability. That means a linear instability in wave vector space because there's a band of unstable modes that you have. And that's quite key that you have this spread out. Now let me take an analogy which might help you appreciate of what's happening. In a thermodynamic phase transition, you have a control parameter, say the temperature. And the temperature you tune externally from, I don't know, 100 degrees Kelvin to 20 degrees Kelvin, and for europium oxide, you will have a phase transition which is a ferromagnetic phase transition to order. But this control parameter, the temperature, is fixed. You tune it. You determine what this temperature is. Okay? It's high or low. It's not variable. It's not a function of time. Only if you change it in time you can do this. It's certainly not a function of space. But here, the control parameter is the density of these proteins, and the control parameter is a dynamic field. So here it's as if in a ferromagnet you would have temperature as an additional dynamic field that's making things magnetic at some spots and non-magnetic at other spots. So here the analog is, at some spots in your system, the system is stable, and other parts it has a limit cycle. That's the analog of phase transitions. Okay, now that you have got this, we can start analyzing the simulation. So this is now with the parameters I showed you, a finite element simulation done by Jakob Halatek at that time. And you can reduce this to a two-dimensional system just to make the plots simpler and easier to digest. So you say you cut out a surface that you have here. Let me switch off the light, maybe. So that you only see the patterns along a line here, and not in a 3D surface, just for simplicity. And then you ask yourself, what is the attractor of the dynamics? Not the fixed point, the limit cycle, but what is the attractor of the spatial dynamics? So can I somehow define a geometric object in phase space that corresponds to the flux balance subspace that we saw yesterday? Well, let's try, and let's see how this attractor that we find depends on the height. And here I come back to your question, right? If you change the height, that basically means I change how much stuff I have on the membrane and how much stuff I have in the cytosol. I change the fraction. That's my tuning parameter here. Now, so here we go. This is now a numerical experiment. And what we do have here is our control space. Now, just plotted differently. Instead of min E and min D concentration, I'm plotting the total concentration of my protein on this axis. And on that axis, I'm plotting the ratio of the two protein concentrations. And remember, we did in control space a bifurcation analysis, and we figured that in this lobe, this gray lobe here, there's an oscillatory instability that you have. We start our simulation with some kind of spatially perturbed initial state, so some spatial modulation of the state that we have, and then we plot here the amplitude of the pattern, basically. That's what I call a state. And that's a reduced representation, right? I'm reducing all of space to the total concentration of the proteins to the amplitude, and the space is reflected itself by a distribution of these blue dots that you have in this reduced phase space as the flux balance subspace that we saw yesterday. Now what happens? So as you start the system, it has a lateral instability. Okay, this is what I said. There's a projection of this modulation onto this reduced space. And now let's look how this thing unfolds. So you have a pattern in real space, and that corresponds to an attractor in the phase space. And you see the lateral instability, the mass redistribution instability, blows up this initial perturbation. This is the exponential growth. And then you do have certain parts in space being part of that locally oscillatory unstable regime. And what you see is they are acting like pacemakers. And these little pacemakers drive the rest of the system. And you see, after a while, this is going into a state which is pretty chaotic. I don't want to compare it to turbulence or chaos. It looks like a chaotic regime, so disordered kind of wave pattern. And this disordered type of wave pattern has its reflection in this rather fuzzy structure of attractor that you have in phase space. Okay? And that's what I would call one way of geometrically kind of grasping of what's going on in a spatially extended system, which is analogous to what we do in ODEs, where we try to characterize things by limit cycles or all kinds of chaotic attractors. So next, we move this thing closer to the boundary. So here you see that the lobe has increased in size, and that just means I have moved the initial concentration of the min E, min D closer to that lobe. And now we do the same game. We start with some initial perturbation, let the system run. It has a lateral instability. The fastest growing mode grows exponentially fast. This leads to a mass distribution instability. Things blow up. You get parts of the system being in the regime of the oscillatory unstable behavior. Well, and now it's much nicer in terms of being more structured. You have here a structure which is kind of a hybrid between a traveling wave and a standing wave. It's a chimera state. That's the language people use in nonlinear dynamics. It's a simultaneous presence of two different types of waves. And what you see then here, the attractor now becomes more focused down. It's no longer that fuzzy, not longer that chaotic, but more like almost a limit cycle, right, in this space. Now, and if you repeat the game, you move this initial point closer and closer, and I don't repeat the simulations. In the end, you'll find a structure in this reduced phase space, which is really a high-dimensional limit cycle. And I feel, or we felt at this time, this is a nice way of putting the results of the system, because it's very much in the spirit of Poincaré, who said that you need to represent things in low-dimensional spaces in order to figure out what kind of attractor you have. And that's precisely what we did here. So I would say this is a geometric representation and characterization of the spatiotemporal dynamics. And what I would love to see, but we're not quite there, is the analog of attractors that we have in ODEs in PDEs. I know this is a big call. It's a big problem, but I think it's worthwhile looking into that. So that's my understanding of the system. That's how I view it. So testing the model. So testing the model is doing experiments, because that connects you to reality. Now, the experiments that you see here are experiments in the group of Petra Schwille, done by Simon Kretschmer, and they actually did it because we asked them to do those experiments. That's a rare event, but it happens. So we wanted them to check this prediction of the theory that the min E concentration has to be less than the min D concentration to see pattern formation. And if you look at this, this is bad. It means you have pattern formation over a huge range of parameters, right? Not just for concentrations where min E is less than min D, but also in the other regime up to 10. So this contradicts all theory. They're all wrong, right? And that's great, right? If a theory is wrong, or a model is wrong, theory is not wrong, a model is wrong, then you learn something about it. You learn that you're missing an essential element in the description of the system. Now, Nigel put it so nicely that if you want to do biology, you have to learn chemistry, or you have to actually take chemistry seriously. Well, we did. We did take chemistry seriously, and we looked into the literature of the chemical studies of these mean proteins. And those are basically structural biology data. And they're not so easy to interpret because they're structural biology data, they're not biochemical measurements. But what we took from these papers and what people wrote about those papers is the following story. So what we figured is that there's not only this switch of the min-D between the ADP state and ATP state, driven by nucleotide exchange and hydrolysis. What we found is there's quite some indication from those data that min-E is also a switch. Okay? And this min-E, well, that's now a more molecular representation of this min-E protein, and it's supposed to visualize that this min-E exists in a closed conformation or an open conformation. And the difference between the closed conformation and the open conformation is that in the open conformation, these little wiggles here, they are the membrane targeting sequences. Those are the sequences the protein needs in order to really bind well to the membrane. They are unfolded, so in a way you can think of this system as a latent min-E state that has a low binding affinity because the membrane targeting sequences are hiding, and that has a high binding frequency because the membrane targeting sequences are actually unfolded and ready to bind to the membrane. All right, so that's a story you have from the structural biology data, and then Jonas Denk in the group actually did a theory where he repeated all these calculations that you see here, and he simply added this reaction on top of it. So why was this crazy? Well, it was crazy because the switch is fast. It's a chemical switch that goes back and forth very, very fast. I told you the oscillations are in the minute time scale. This is in the sub-second time scale, so it's super fast. A person who does chemistry would tell you, you can integrate that out. It's not important because it's equilibrated before you look at it. So you can regard it as an effective degree of freedom, and you disregard the opening and closing. But we still said, let's try and do the analysis, because after all, this is not a well-mixed system that you have. You have a membrane, a very small layer of gradients close to the membrane, and you have chemistry in a spatially extended system. So let's take this really seriously and work out the details. So he did, and this is Jonas, this is the hero, and those are the equations, and you should no longer be scared by those equations, because they're just a minor modifications of what you actually already saw on the blackboard, and if you're not too tired tonight, then I ask you as an exercise to kind of look at those equations and derive them for your own. It's not hard, it's really not hard. And the prediction of that theory, the basic prediction is, and I don't go into details, is that this double switch, this min-D, min-E switch pair, is essential for the pattern robustness. Because that's what they saw in the data. They saw patterns over a very broad range of min-E to min-D ratios. And that means biologically that these patterns are robust. If you have different expression levels of min-E and min-D, if the ratio changes, the pattern doesn't care, it's robust against those changes. So it's not just a nuance, right? It's not just a minor addition to the model. It's a module that explains a feature, namely robustness, which is not there in the original model. Yeah. It's typically very, very fast, and of course we put some parameters into the model. Again, these parameters are not measured, unfortunately, but it's very, very fast. It's something where you wouldn't, if you were to consider it as a well-mixed system, you wouldn't even think about leaving this in the model. Those are, again, there's now an additional list of parameters, and the sport is, and this is not easy, to infer those parameters by comparing your theory with the data. And this is research that has been going on for many years, and many different people tried, and this is converging slowly, I would say. It does converge.
So if this is going so fast, could one just put this on quasi-steady states?
That's what I'm saying, no.
Because?
You'll see.
Okay. Okay, but so you're going to explain how we can understand why
Yeah.
this is giving... Okay.
Yeah, I explain. It's very weird. I mean, I fully agree. My first reaction was, forget it. This is not going to give an effect. But the point, I think, which one has to appreciate is this is chemical reaction kinetics in space, and concentrations are not constant. They have profiles. Okay, that's the key. But we'll see. So now, how do you check? And that's where we convinced Simon Kretschmer, at the time PhD student in Petra Schwille's lab, and they really were willing to say, okay, we'll check your theoretical ideas. So what did he do? He has, of course, the wild type system, which is this. And then he can make mutants. And by the way, wild type for me means it has a switch, and it does have these little tails that are the membrane targeting sequences. Right, so this is a little unusual. It's physics, but you have to worry about biochemistry. Now, what he did, he made a mutant. They have names, don't worry. It's called L24N, and has no min E switch. So it's able to modify the proteins such that they no longer open and close. Okay? There's a whole story on its own. It's their business. We trust them, what they do. And then he can actually cut off the membrane targeting sequence. So he can separately check the relevance of the opening and closing and of the membrane affinity of the min proteins. Now here is what he found after a while. So this is a PhD. It's a lot of work. Comparing the different situations. So we have the wild type, we have no membrane binding, we have no min E switch, and we have a mutant that has neither a min E switch, nor does it have membrane binding. So what do we see? The wild type is, as we started out with, this robust pattern formation over many different ratios of min E and min D. And if you switch, if you remove these guys here, the membrane targeting sequences, nothing happens, right? With nothing, you have to be careful so that the range of patterns remains the same. What changes, after all, is the length scale of the patterns, okay? So there's an indication already there's something nice about or interesting about the binding of the min E. It seems to be another module that's relevant for the length scale that you have. Well, if you remove the switch, victory, right? Because the switch was not present in the original model that we call the skeleton model, and there you do indeed find that you have to have a min E concentration less than min D to get pattern formation. It's a nice confirmation of the theory, and if you then remove, in addition, the membrane targeting sequence, again, the range doesn't change, but the length scale of the pattern changes. So in short, you can map theory and experiment concerning the fact whether the patterns are robust to ratio changes of min E to min D. Now here's my explanation for what is the mechanism. There's math behind it, but let me wave my hands because I want to give you an intuition for that. So now you have to remember of what I said about the mechanism of min oscillations to begin with, so in the skeleton model. The mechanism was that you needed to be able to accumulate the min D proteins on the membrane before they're kicked off by the min E, right? Because otherwise no pattern forms to begin with. That's what you have here in this case. And now, what happens when you have more min E than min D, but you have this switch between a latent min E and a active min-E. Okay, let's start from the right-hand side, because the min-E's are made from latent to reactive by the min-D's on the membrane. So initially, there's just latent min-E in the cytosol. Okay? And they're not able to bind because they're latent, right? They have their targeting sequence is hidden away. So during that period, you can have as much min-E as you like, because it's not functioning. It's hiding in the latent state. So you can have accumulation of min-D on the membrane. That's the point. We have something that's not active yet, so you can go on the membrane. Once you are on the membrane, well, then you start switching all the latent min-E's into reactive min-E's. Okay, so you activate the min-E, and once you activate them, well, you activate them in a particular region, you activate them close to the membrane in a tiny layer, right? So this is a spatially restricted cloud of reactive min-E proteins right in the vicinity of the membrane, and those then, they are able to kick off the min-D proteins. Okay? And that's why you can have, despite the fact that you have a fast reaction, you can have, because it's spatially localized, you can have a pattern-forming system in that case. Okay? That's the hand-waving explanation. And here's my cartoon, right? Min-E is kind of hovering over the surface, and that's the, I called it hoverboard mechanism, but that's more of a joke. So hope you got the idea that a fast chemical reaction can have a effect on pattern formation if you restrict it spatially in the vicinity of the membrane, and when you have the sequence of reactivating inactive min-D.
If I were to ask, forget about the biology, but how would you change in the most parsimonious way your first model to be able to effectively get that effect without having to put in an, like, could you have done this?
No.
No. Because of the spatiality.
Because of min-E has to be less than min-D, independent of whatever you do to the original model. The original model, whatever you choose the parameters.
Even if you would like put up like some cooperativity somewhere for it to bind more for itself.
Yeah,
so, because effectively it's doing that, right? It's giving a cooperativity for the...
Yeah, you can always cook up some nonlinearities, but that's something we didn't want to do.
No, it's not that it's just for me to understand, to universalize this in...
I think at this point in time, we really made a bet in the sense that we saw the min-E switch in the structural biology data, and we took this serious and implemented it, and it works in vitro. Yeah. Is there another way of doing it? I mean, I cannot answer this, right? Yeah. But this is a very convincing way of explaining the data, because the mutation analysis
definitely, don't get me wrong, it's beautiful and it's definitely the way. Yeah. I was just thinking, like, if I could take a message from this, so if I see another system which might not have implemented it in this way, I would be able to have a search image for what effectively would be necessary.
Very hard.
Okay.
Very hard. So I find this super hard to answer, because those biochemical reaction networks are complex. And I think I mentioned it in my first lecture when Lila Vailan did the experiment on the CDC42 budding yeast system and deleted BEM1. The question was, what happens to that biochemical reaction network? And what she found experimentally by biological evolution is that it reduces to the min system. And so the question backward, how do we evolve a system to have certain additional modules that give rise to robustness and so on and so forth? It's not clear. But having said this, the hand-wavy explanation I gave, this is very generic. So as soon as you have something that can hide away and it's activated only by things on the surface, you have a mechanism that broadens the parameter range for which you get pattern formation. So, yeah. Yeah.
Yeah, I've got two questions, I guess. Yeah, about the fast protein switches. So suppose that the protein switched, with a period of 10 to the minus 24 seconds or something like that. Would this mechanism still work?
It of course breaks down,
right?
Because there's this. You have to, if you do it more carefully than the hand wavy part of it. There was this, cloud, and there's this length scale,
right?
And that's the point, right?
So the frequency of the protein switch sets the length scale of the localization.
Yeah.
So then okay, great, thank you. So then my question is, then doesn't discreteness matter on that length scale? In other words, could stochasticity be the thing that is actually helping to maintain the pattern robustness?
Stochasticity, we have looked.
Due to discreteness.
I know what you're talking about. I mean, number fluctuations and the number fluctuations.
You're in a very small area, so the sort of, you could say the PDE description now breaks down. I have to seriously worry about a few protein switches that are in the reaction volume. Yeah.
There's various elements to this answer. I mean, the min E and min D proteins, they are abundant in numbers. So abundant means thousands.
Wait, outside, yes.
Thousands that you have.
Yeah. But just at the reaction, just at the site where the protein switch is mattering in this little layer.
I mean, this is something you can look into. At the moment, I can only give you kind of my intuition on it. I would not think that this gives rise to the robustness of the pattern, but we have not analyzed it, so I cannot tell you, really. Yeah.
But it's very
stochastic dynamics there is not so easy because you have to run Gillespie for that. Yeah.
But I mean, is it possible? I never thought of this before, that just because you've got stochasticity in this one localized region, that can then somehow renormalize in some sense the parameters for your bulk description, and that's what gives you this unexpected robustness.
You would need to have a different explanatory story that gives rise that this broadens the parameter range. So the latency is my explanation, switching to activity, and you would need something that I think more on a deterministic level can go from latent to reactive. If I think of stochasticity, my intuition would tell me you have that, but not in such a systematic fashion. You would have it all the time happening. Yeah. But anyway, I cannot answer the question in detail because we haven't done the analysis for that.
In the regime where you see the patterns where you didn't expect to see them, could you show your sort of slide again?
Those are very strong attractors, if you mean that. So those are pretty regular patterns that you do have.
Yeah.
Yeah. So, and that's my intuition, Nigel, behind those systems. There's attractors that you have from the nonlinear dynamics. They are pretty strong. And now if you think in terms of a Kramers-Moyal type of mechanism, then you get a cloud around those attractors. And in your system, I guess you tell the story tomorrow, you do have this spiral, and the noise is creating a new type of limit cycle, right? That's basically there. And this is distinct here because you already have a limit cycle, and you're perturbing around the limit cycle in a metaphoric way. Yeah. All right.
So one last question, since you mentioned that. Did you look at the power spectrum for the fluctuations of the signals of the min D, min E?
No. The people that did those studies didn't do this.
Okay. But the data still exists, or you just have those images?
It's fluorescent data.
Oh, okay, yeah.
So it's very
hard,
difficult to quantify. And there is fluctuations coming from attachment and detachment. And there's other kinds of fluctuations. So it will not be easy from the signal to really infer specifically on certain types of correlation functions, which you want, right? You want to have a correlation function for the min DE bound to the membrane, the min D bound to the membrane, or those that are in the cytosol. These are already tricky for the actual concentrations, and it's even harder for measuring correlation functions. Yeah. But that would be, you know, a theoretician's dream to have those kind of correlation functions available to you, because they would constrain our models even more than they can do it now from the mean values.
Yeah, thanks.
Right? Please ask.
Can you explain again how does the mean E switching works? Like, why is it not active in the cytosol?
Yeah, it seems from the data, again, this is what we read into the structural biology data, that only the active min D, and the active min D is bound to the membrane, is able to switch the min E from the latent to the reactive state. It's not a switch in the sense that it's a stochastic switch that opens and closes on its own. The switching is facilitated by the active min D.
So the active min D is formed in the cytosol, right?
No, it's so to say only in significant numbers on the membrane, and even if it would be in the cytosol, to find each other in the cytosol is very hard.
But
it's
if
a
you have
three-dimensional volume, and the concentration, that's the volume concentration, Veronica asked about this, and to the first passage time problem of a chemical reaction to actually happen in the cytosol is really, really low. So that's why also all the chemical reactions you saw, they all happen on the membrane. In the cytosol, only trivial stuff happens, namely nucleotide exchange, that only requires an action that happens on the protein itself. A reaction between two proteins is not likely to happen anyway in the cytosol.
Even
because
if
of
you have so much
dimensionality.
min E?
Yeah.
Like if you have like 100 times min E to min D, wouldn't you expect it to happen?
You have
like
to think this is a gas. They're far apart. And you have to calculate the chances of them meeting, and that's just low.
Okay.
But I would say it's a physical explanation. All GTPases that are only active on the membrane structurally. So this is the reason why probably has
Yeah.
evolved to be so, but it is the case. You cannot do your job molecularly if you're not inserted into the membrane. And when you say going from active to inactive, it means you do something. You actually you performed something. It could be signaling, asking phosphorylating something, hydrolysis in this case, you're doing something, calling a vesicle. There's some action which is performed, so which will be, things will be done, like the cell does something, right? And then it becomes inactivated. Seemingly it can't do two or three things. It actually does one job and is inactivated. By doing the job, the switch is done. It doesn't just become inactive by itself.
Yeah.
It undergoes a step. And this is for this system, for any system. So this is the good news. If you learn this, all of the rest is the same principle. But you can be inactive in the membrane. That you can be.
Yeah.
You
Yeah.
have to be inactive to become activated.
I remain curious about the patterns of the experiment that you showed, because for the high ratios, they look more or less like Turing patterns. They are fixed, no?
We are using the word Turing pattern here in a very loose sense.
Yeah, of course.
So they look like periodic patterns.
Yeah.
Let's call it this way.
Because previously they were oscillatory waves.
So, yeah, it's not time resolved, right? So you don't see the time resolved patterns that I showed in my simulations. And the simulations were performed in a computer simulation to make points about attractors. And now you look at experimental data, And there you find also in the experiments, you find traveling waves, you find spirals, you find target patterns, you find labyrinths, you find all kinds of patterns depending on the parameters. And it's of course a very, very interesting question to ask: what determines the length scale? And what determines the time scale of those patterns? And what's the biophysical or biochemical mechanism behind a length scale, behind a time scale, behind a traveling wave, behind a standing wave? And I'll come to that in my discussion of the in vivo system. And at present, we are asking those kind of questions about the in vitro system as well. This is, not finished experimental studies of Kees Stekker's group, where we compare the simulations that we have with the in vitro data, really trying to answer the questions you are asking. So here we were kind of putting ourselves blind to the actual patterns. We only wanted to know what's about the robustness. Yeah.
Yeah, but I want to aim at, if you add this ingredient of the switch in your model,
Yeah.
and now you do your numerical simulation
Yeah.
for these high densities,
Yeah.
you observe still the traveling waves?
Sure.
Or they become kind of stationary like these ones?
Depending on parameters, you can find all kinds of patterns.
Okay.
And, it has become a business of looking at patterns and claiming explanatory value by seeing the same thing in data. This is a very dangerous track you're going. You have to, and that's what we do, change conditions in the experimental system and see how correspondingly you have a change in the theoretical model that matches with a corresponding observation in the experiment. So it's really about quantitative understanding as well that you have to be careful about. Okay? Please.
If the parameters matters to the kind of patterns that arise from your system, when you are studying a new system with little knowledge about the parameters, how do you deal with that?
Very good question. And this whole story goes now on for, I would say, 10 to 15 years that we and others have been studying this system. And because you don't know the parameters, you have to come up with the parameters from fitting the data. That's what I told you, right? And ideally, you would fit the parameters once and for all, and then change the experimental conditions, and your theory still remains valid. That's the ultimate goal. And at the moment, I think we're actually there, that we have one set of parameters, and with one set of parameters, we can explain the in vitro data, and we can explain the in vivo data. With minor modifications, because in vitro and in vivo is different in terms of diffusion constants, because the cytosol and the membrane have different types of viscosity. But basically, these different sets of experiments are explained by the same set of data. That was not true, not at all true, when we started out with that. So the initial theories that we had gave, as you saw, the wrong answer for the min E to min D ratio. And they gave the wrong answer about the time scales and the length scales. Okay? But that's how it goes if you really want to go to a molecular model and be serious about it, to make the molecular model molecular, so it really explains from the micro to the macro, right? And that's a dilemma everybody faces. And let's say for the min system, we were now lucky to be at a point where the theory is quantitative. For other systems, we never may be there because the molecular complexity is just so high that you cannot achieve this mapping between the macroscopic physics and the macroscopic behavior. Yeah. It's one of the key challenges, I would say, in modeling biological observations. And I mean, Veronica, you alluded to this very nicely in your introductory talk. There's a lot of unknown unknowns that we don't really know. Yeah. Other questions? All right.
Sorry. Here.
Please.
So if you squeeze the cell partially and
Yeah.
abruptly, can you go away from the pattern formation region? Because you are changing the,
Changing the length scale, yeah. So answer is, as you change the length scale, the length of the cell, or the aspect ratio of the cell, and here we didn't, this is in vitro data, there's no length, right? This is a huge two-dimensional membrane and a certain volume on top, but if you were to do this experiment, and I'll come to that, in a real cell, the aspect ratio and length of the cell changes the pattern. Okie doke. So the final frontier is trying to actually look at this in a way that you do experiments in a living system. And here there are several heroes in the story. The student Ren here in Soo-Kyun Yoon's group, he started that, I don't know, 6 or 7 years ago as an undergrad to do these experiments. And it just came up because Soo-Kyun and myself were meeting, and I was kind of suggesting to him we have this nice prediction about the mini switch, but we only have in vitro data from Petrashvili. Can you check our theoretical prediction in a living system? Can you actually really do this? Okay. Well, and then he said, I can manage, but it took quite a time to actually realize that. And what they do is what I cannot explain to you quite well, but they change the transcription of the min, C, D, and E proteins in a controlled fashion. And to make a long story short, they can make an in vivo phase diagram varying the min E concentration in vivo in the living system and varying the min D concentration in the living system. That's what they can do. Of course, there's all these questions. How do you make sure how much you change the min E and min D concentration? How do you measure? How do you quantify? How do you put a label on the min E or the min D concentration? Okay, but there's lots of issues, but basically they can change the min E and min D concentration. And then, you know, look here at the lower figure. You can have a priori, just without looking into any theory or any data, you can a priori have different types of scenarios in the biology of the system. You could have a fine-tuned oscillation for the proteins, such that only in a very small domain of min E and min D concentration, you actually have oscillations of the min and D proteins between the two cell poles. Could be, you know, nature through evolution could have found a spot, and this one could be, you know, fine enough to make the system faithfully divide into daughter cells. What you could also have is that you have oscillations that are over a very, very large range of parameters, and the system is super robust against any kind of fluctuations that you might have. But then you could pay a price because you would make this large parameter regime, but the system would not be resource efficient. It would not produce just the right amount of min E, min D proteins to make those oscillations and not more, right? Or third version is you do something like that, where you have a resource optimized regime which is at a boundary of this min E, min D phase diagram, where the wild type is and where you have the oscillations, but at the same time you're actually resource efficient. Okay? And there's lots of reasons to argue for that, to something that through evolution is optimized to use resources in an efficient way, to produce the biological function it needs to produce. That's a priori possible. And now let's see what the data say. So this is what you just saw, sorry. This is what you just saw. Come on, doesn't like me. On the right-hand side, that's what I explained to you. That's this latent min-E, acts as a reservoir that buffers high min-E concentrations. And now when Henrik did the simulations in a setting that is confined to the spatial geometry of a cell, what he finds is that, again, this robustness should be also valid in the in vivo system. Without the switch, there's only this narrow regime where you have pattern formation. With the switch, you get a very broad regime where you do have pattern formation. So here's now the experimental story. And that's figures that have been replaced by now by new figures. So this is part of the review process. But anyway, I show you these figures because they basically tell the same story. So what you see here is the min-D concentration in 1/micrometer cubed. Here is the min-E concentration in 1/micrometer cubed. You do have here a range of different concentrations they measured. So you see here these white spots, the blue spots, etc., and then these differently shaded regimes. And in all those regimes, they observed pattern formation. But they also observe regimes here where you have no patterns, right? Here is no pattern, and there is no pattern. And the reason for no pattern is different. In one pattern, you just overexpress certain proteins that you suppress the pattern formation. In the other regime, you have not enough proteins to actually kick off the pattern formation. And once you have pattern formation, you can classify the things that you see. You can classify that some of these patterns, like the violet regime that you have here, shows standing waves. In the regime that is kind of bluish, here you find traveling waves. Like in standing waves, this is a chymograph, and you should read this like so. Here you have time to the right, and that's the concentration profile along an E. coli cell. So you have concentration of the min-D proteins at the poles. Then it's focusing in the middle. It goes to the poles. Here in the oscillations in the normal cell, you have back and forth oscillations. And here in traveling waves, you see there's a traveling wavefront that you have, which goes from one end, one cell pole to the other cell pole. Yeah, very good eye. So because of the different expression rates of the min proteins, something else happens to the cells, that they actually change their shape. And this is a link between the patterns that you form and cell division that happens in the system. So what they observed experimentally is that in these various parameter regimes of the min-E and min-D, in some of the regimes, and it's difficult for me here to see, so if you're here, this is the normal oscillation regime, also there, also down there, and in those regimes, the cell grow longer, actually. They grow in size such that they're larger than the wild-type cells.
But they have to start small, right? Or do they just...
No, they have to start small, and then they grow, and this is the final state where they have grown to a saturated length, and then they measure the data at this saturated length scale. Yeah? So you see that the experimental data on their own, without any kind of interpretation, they do have the oscillations, they do have a very broad regime where they do find patterns of different type that's linked to the size of the cell because of the biological processes that happen, and you find oscillations, standing waves, traveling waves, and no patterns. So the system is actually robust in the experimental system. It has a very broad parameter regime where you see the patterns, and if you look at the wild type, and here there's slow and fast. It's another nuance, because if you really properly grow the cells, you have to worry about the growth conditions and have to be careful about that. This is under different growth conditions, slow and fast growth condition, and in both cases, the wild type is actually found in the lower tongue here of this robustness lobe in the min-E, min-D parameter space. So that indicates system is resource efficient. It tries to kind of minimize to that regime. It could also be here, right? It could do the same job up there and show oscillations. No, it's here where it shows oscillations. And you now have an interesting system that you could potentially compare with theory, right? Yeah.
Sorry, so when you empirically measure the min-E concentration and the min-D concentration, you do that in the end. You grind the cell and measure it, or is it something that you're controlling in experiment?
No, you control in the sense that you change the expression levels.
Okay.
But then you have to measure of what actually is expressed. You cannot engineer such that you know from the engineering what the expression level of the min-E and min-D is. You have to then look at your system, you look at the fluorescence of the min-E and min-D, and then you have to find a way which is convincing that gives a number to the min-E and min-D
so
concentration.
So it strikes me that min-E really varies here, orders of magnitude, but min-D is
varying here in this range from 10 to, you see here the data, 10 to the 2 to 10 to the 4, 5-ish, right? So over quite a range of values.
Okay. So transcription, although you're having a longer cell, you're somehow compensating by making more proteins.
Yeah, you have to measure how much do you have then.
Yeah.
What does the gene regulatory mechanism give you?
It's so much information.
It's a lot of information,
Really cool,
So I think you have to digest it and look at it, and I try to focus your attention just on the fact that there is a phase diagram. There's lots of very important and crucial details of how you measure the min-E and min-D concentration, etc. There's a lot of experimental scrutiny that you have to be careful with, and Suk-Yun Yoon is a master in this, very, very detailed in how he makes these kind of experiments. Okay, and there's a whole story to it which I possibly cannot tell here in this talk. Okay, so I hope I got the message across. It's a broad regime. Robustness seems to be something that's found in vivo as well, and you have different types of patterns, but they are now linked to different length of the filaments. All right, now we come in, or Henrik comes in. Before that, we have a question?
Yeah, is there a reason why there seems to be like an optimal min-D concentration for a wide range of min-E? Like, does that come from the model anywhere that you could, like?
What do you mean?
So a bacteria, to be able to divide, wants to be oscillating. So we would be looking at like the turquoise area, and for that, it's pretty much like around a very narrow min-D concentration until it gets to that other min-E value.
So this is the wild type here?
Yeah, but I mean, like the green shading is basically in a line until it gets to like 10 to the...
This here?
Yeah, all of that.
Yeah.
That seems to be like narrowly around like a particular value in D. Is there like a reason why that would be?
Well, the reason you can say when you now use a theory and you try to explain these data, would the theory also give you that? That would be a reason. Another reason would be I have a hand-wavy explanation of why this is so. I really don't have at the moment. Okay, but there's a reason. This is encoded in the biochemical reaction network, and reason can also be evolutionary reason. I have no idea, right? Evolution happens in those systems, but to have a post-the-fact evolution story, you can cook it up, but I'm not so sure it makes sense. Yeah. Right. Other questions? All right. So now, once you have that, you have to compare, or you can, if this really works out, you can compare with a corresponding numerical computational model. And this is now basically the same model with some modifications as the in vitro model that we had. And now you have to fix the cell length, right? Because you want to compare apples with apples and not apples with pears. So they had to do something to the E. coli cell to fix the length. So you work with the apparatus that determines cell length and cell growth, and they can manage to make cells that are all of the same length. And with those lengths, we then compared the numerical simulations with. So these are now for cells of a certain length, and hope you can see the scale bar. The scale bar is here 5 microns, and in time, it's a very bad picture, is 120 seconds. Okay, and you see not so well, it's actually a pretty bad resolution, that in these fixed length cells, you basically have different type of patterns. You have standing waves and you have traveling waves, and they're much clearer, of course, in the numerical simulation. Here, as you look at the cell length and you look at a kymograph where this here is time, you see certain points where you have these body patterns, and then you have traveling waves. These are indicated by things running down here in time. Here you have a standing wave. That's again a traveling wave. Okay. And now where do we find, where does Henrik find traveling waves, and where does he find corresponding standing waves? And the color code that you see here is comparing the different types of patterns that you see in the experimental data and the patterns that you see in the computational simulation. And there's a correspondence between the two, which is pretty quantitative, right? This is really comparing things at the scale of the concentration of the proteins as far as they can be measured. You can also fix the concentrations to the wild-type concentration and then measure relative to the wild-type concentration. Then you're a bit safer, because then you just talk about relative concentration and not absolute concentrations. And that's, in the end, how the plot was made in the final version of the publication. So if you measure with respect to the relative concentration of the wild-type, there's a one-to-one correspondence between the patterns, the range of the system showing patterns, and the type of patterns that you have. And that we were already quite happy that we have this nice correspondence between the data and the experiments at the level of a pattern. Challenge, as always, what about the length scale of that pattern? And that's something we didn't really hope to see at the moment, honestly. So at first, we didn't really look at the length scale because we were like, it's a bit asking much of a theory to get the length scale right. In the end, we did anyway, or Henrik did anyway. And here you see the data from the experiments. I apologize for the bad resolution. And this is the wavelength here as a function of the mean E concentration and as a function of the mean D concentration. And what's striking are two things. If you look at it with the eye of somebody doing a model and comparing with experimental data, you strike victory, because this compares quantitatively with the experimental data. What's interesting, though, if you look at the data, is that there is no variation of the length scale. This length scale is 10 micrometer, as you see it here, and it's pretty much flat, independent of the concentration of the proteins. Okay? And that comes out from the analysis of the data, and it comes out from the analysis of the computational study. And it's a very striking observation that somehow this system has found itself to be length regulated such that the length is constant. And that's reassuring for the biological system because the robustness of a length scale is essential for the function of the cell. It needs to divide at midplane. And if it would not be robust to variations in the concentrations giving rise to the different length scale, then it would not be faithfully having a system that correctly gives the function of the cell, which is dividing at midplane. You can also see here that there is the wave period. So this here is experimental data. The dots, the black dots, are numerical data. And this here is actually the analysis from a linear stability analysis. And the reason we can do this is that the transition, it turns out, is actually subcritical. And if you have a subcritical bifurcation, then you can do linear stability analysis, and the length scale and time scales are really those that you find from linear stability analysis as in a Turing pattern. And that also, as you can see, the numerical data agrees with what they see experimentally. So to make a long story short, it's quite striking at the moment how accurately the model represents the experimental observation in vivo, in vitro, at the level of patterns, at the level of wavelengths, at the level of timescales. So I'm pretty sure that we are not far from a really molecular model for the MIN system, where we can explain things not just qualitatively, but actually quantitatively. There's one more thing. There's min-E persistent binding as well, which is not there in the model. This model here has these features, right? It has min-D activation, min-D attachment recruitment, dimer formation, hydrolysis, the min-E switch. But here, you see there's something else. You could actually have hydrolysis, and this min-E is actually stubborn because it has its membrane targeting sequences and just doesn't let go and stays on the membrane for a while. And we're presently studying that, and it turns out that this is the module that determines the length scale. And you saw it already from the data that I showed you when we were changing the min-E binding, it changed the length scale of the pattern. So you have kind of a modular picture that comes out from the analysis. And this is the summary. So the min-D, min-E basic model, we call it skeleton model. I don't know how the name came up, but that's the name we gave this thing. This explains the basic mechanism. And that model was first written down by Casey Wang and Ed Wingreen. They came up with that. We analyzed it. Other people analyzed it. We're all happy that this model works. But in the end, if you look at it closely, it doesn't work quantitatively. And one major step, I think, is the min-E switch model that we introduced. This min-E switch model explains the robustness of the min oscillations to variations in the min-E, min-D concentrations. So it's an important module. It's the robustness module of the MIN system. That's how I would call it. The third one is this persistent membrane binding, the stubborn min-E protein that doesn't want to let go. And that we find is the module that is responsible for the actual value of the wavelength. And that is in the simulations that Henrik performed for the in vivo system already. And now we are testing this with an in vitro system. So we go back to an in vitro system, this time with KS-TECA, to check whether you really have a variation of the wavelength with that. And the reason is it's much easier in an in vitro system to change the wavelength. In an in vivo system, you're always bound by the geometry of the cell, and you cannot really vary. And from that, you see that there's a real great value that in these studies you do have the reconstituted system and the in vivo system, and you can go back and forth between the two. Finally, there is an open question. What's recruitment? I mean, there were some questions about what's recruitment. I must say, I don't know what recruitment is. It is this: a reaction term where you have a bimolecular reaction between the min proteins on the membrane in the cytosol. But what's the molecular mechanism behind that? That's unclear. I would love to learn what this is. All right, I think with this, I'm at the end for today, and you must be tired. So thanks for your attention.
So do we have any question?
Do we know?
What drives recruitment?
We don't. I think I mentioned it before. I have a model which was developed by Andrei Gojchuk when he was a PhD student in the group. And Andrei was developing a model which is based on mechanochemical coupling. So it's based on the fact that as you bind proteins, they kind of prepare the curvature of the membrane in such a way that the next protein is finding the right curvature it likes to bind to. Okay, so and there's indication from experimental data that min proteins create a spontaneous curvature at a membrane. That is a possible mechanism, but if you look at E. coli cells, their membrane is stiff as hell, right? This is a bacterial membrane. And so it's not so clear whether any kind of deformation and mechanochemical coupling would be a possible mechanism for MINDI recruitment in an actual E. coli cell. But I like the model. I like the idea that there's physics which determines chemistry and gives rise to recruitment. That's as far as I can say something on recruitment.
So on the first day, you had a feedback term that was m squared.
Yeah.
And now I can see that you have like linear in m and now m squared.
Thanks for that question. So on the second day, right, with the two-component model, these models are cooked up. They're coarse-grained models in a reduced space of just one protein with two conformations. So I'm hiding all my ignorance about the biochemistry in a guess about the function and the nonlinear shape of that function. And in reality, what I would need to do, I would need to take this model, which has true biochemical reactions, bimolecular biochemical reactions, and would have to coarse grain that model in some way to eliminate variables to then come up maybe with a two-component model. Okay? So anytime you see nonlinearities, this is a modeling step, which is good and fine because at some point we just don't know all the molecular details, but it's distinct from a molecular model like so. Yeah.
Thanks for the talk. I'm guessing, how did you made your quantitative predictions? Because I see that the model has several parameters. So did the experimentalists measure all of them, or?
No. I'm not a strong enough personality to bend the arms of my experimental colleagues to measure those parameters. At some point in time, there was the hope that you could actually by measuring the mass of the proteins, and there are methods to do that, to identify which of the proteins you see, and then measure the biomolecular reactions for those. And there's some very nice experimental work on that. Problem is the min E is too light to actually see this in this kind of data. So it's an open question to measure these biomolecular rates. It's just not known. And what we have to do is an inverse problem. The inverse problem is you have lots of different data, right? You have in vitro data, you have in vivo data, you have them at different concentrations, you have them at different bulk height, and by now there's a huge range of data that you have for the MIN system. And over the many, many years, we have converged now to a set of parameters and model structure that seems to be robust enough and quantitative enough to explain the data. But it's ultimately an inference problem. Yeah.
So, well, two quick questions just to understand this. So you very nicely show the robustness, right? Independent of your min E concentration. But when you said, oh, my model is robust, it's robust to this, right? But it is then, you can tune the wavelength with the min E
persistent binding.
Persistent binding.
Yeah,
right.
that's true.
So, and how sensitive is that? So you can vary then
You can vary it quite a bit, not over huge ranges.
Okay. But that's your tuning parameter.
The tuning parameter is, you know, what fraction of the min E proteins remains persistent and what fraction goes back.
Perfect, yeah.
That's the modeling parameter, and as a function of the fraction remaining, you can tune the wavelength.
Adjust it a bit, okay.
Yeah. And it was a priori for us not clear what is the biochemical reaction that is responsible for the wavelength, because from such a complicated reaction network and the nonlinear dynamics, as you know, it's a problem on its own to find the length scale. It's not a simple Turing problem where you would read it off from dispersion relation. It's a problem where the length scale that you find is a non-linear length scale that emerges after the nonlinear dynamics has set in.
Yeah, that's super interesting. In plant cell, we have this phenomena that they make a root hair at a certain patch, and when they mutate the way that you bind to the membrane, so you know, actually you can make many different patches, and that was very confusing, right? Because it's not, you're not changing any of the other kinetics, it's just the. But then the next question I had is, so very nicely the model recapitulates the patterns. Have you tried this on the cell itself with differing lengths? Because experimentally you saw that a small or a longer cell could give you different characteristic patterns. Yeah. And so that sort of gives the idea that it goes through something more non-local, right? Do you have ideas how your model might work for
cells of different size and different aspect ratios?
Yeah.
Yeah, we have done that study. So Kees Dekker's group has squeezed E. coli cells into different containers of different lengths and different aspect ratios.
What you showed on the second day.
What I showed on the second day. And there's also a theory story that comes to that. And in the theory analysis, we compared the different types of patterns that he sees, or they see in their experiments, with the different types of patterns we see at that time still with the skeleton model.
And but not for Sukyun's data, so you didn't?
No.
Okay.
For Sukyun's data, we at some point decided we need one length where we want to compare things.
So you didn't?
Okay. Yeah, and we decided on one particular length, and there we did the tuning of the parameters, because you still need to do this, right? This is not, you can not just take parameters off the shelf, and then we fixed it for that. Okay. Because in the end, you know, if you change the length, you change the physiology of the cell, and when you change the physiology of the cell, you cannot just take the same parameters.
Fair enough.
Yeah. Done.
Any last question?
I don't have a question. I have an announcement to make. Actually, I have a question for the audience rather than for Erwin. So tomorrow I have my hands on, and I actually wanted to give you some code, and I want to ask you a question for this. I actually prepared codes in Python, in R and in MATLAB. So just a question here. Who here would be able to run Python code? That's a lot of people. Who here would prefer MATLAB code? Great. Well, any R users here? R? Well, one. Great. Hey, you must be an experimental ecologist or something like that, or not? Maybe not. You also? Yeah. A little bit. So I think that Python code is favored, but also there's the MATLAB